So hello everyone. I'm recording this video in preparation for our evening session on this Wednesday, the 10th. And rather than us having a live online session, due to few other factors that have taken place, what I'm going to have to do is provide you this session in an asynchronous manner. So I'm providing you this recording so we can go through the material that would relate to the lab number 9, which is all about inheritance worksheets. So earlier on this week, we talked about the genetic inheritance of characteristics. And on this lab, what we will do then is apply that knowledge. So even though we don't get to meet in the same time synchronously, I still wanted to have a session with you where we go through the material. So before we get any further, I think it's important that we just kind of remind ourselves of some of the very core concepts that we saw in the past 2 chapters. So first of all, whenever we're talking about genetics, we're talking about the genetic makeup of an individual. So individual genetics are determined by the material that we inherit from our parents, and that was of course in 50-50 ratio. So you inherit 50% of your DNA from one parent and 50% from another parent. And we had a look of that process, how that actually happens on a cell level in a greater detail on our earlier chapters. But at this point, all we need to know is that we inherit 50% from one parent, 50% from another parent. We have also already earlier on these past several chapters discussed about the concept of alleles. So for each trait, we can have 2 forms. For each gene, we can have 2 forms, and each individual carries 2 of those, but you will inherit one from only of your one parent and another one only from another parent. So it's really 50-50 chance that which allele from each of your parents do you end up inheriting. So this process aligns with what we discussed, that in the sexual reproduction, we want to really add up those chances that we get a random mix of genetic material for this new individual and hoping to hit that genetic jackpot. You remember that I showed you the picture of that raffle machine. We really want to come up with a combination of alleles, combination of various genes that makes that individual especially well adapted for surviving, being very successful, and through that being an attractive mate, and in that way passing those genes onwards to the next generation and next generation. And that is exactly how the evolution, the concept of evolution works. A couple of other important terms that I want us to discuss about. Let's have a look of those. So we mentioned the concept of genotype as the genetic makeup that you're made of. And I've taken now here one trait, and that trait is symbolized with letter G. So you can have either 2 alleles that are both capital G, one capital G, one lowercase g, or 2 lowercase g's. So all of those would be variations of different potential genotype, different potential genetic makeup for that trait. Well, we are going to use a lot of this genotype and focusing on which kind of combinations we can end up with, and starting from different kinds of combinations. But really, ultimately, what we're interested in is the phenotype of an individual. So phenotype is what we actually see in that individual. So the traits that appear. So for example, we discussed about things like hairline, we could discuss about earlobes, we could discuss about when we look at the pea plants, their color, whether the seeds are wrinkly, and so on and so on. So all of those phenotypes that we see, all of those traits that we can observe are dependent on the genotype that that individual will have. However, at this point, I do want to emphasize that there are sometimes certain alleles that a genotype of an individual ends up carrying, but we might not see. So that's usually the case when we have the capital letter, lowercase letter. What we will see is only that dominant capital letter allele. We won't know what's going on with the second one because that's going to be always masked. So kind of to expand on that concept, we need to remind ourselves of the concept of dominant and recessive alleles. So dominant alleles were the alleles that we always end up seeing, regardless whether there's two of these or just one of those. And this is because as long as we inherit even just one dominant allele, that is what we're gonna see. So a single one dominant allele will mask the expression of all other alleles for that trait. So whether it's capital G, or capital G, lowercase g, the outcome that we will see as a phenotype will be the very same. Well, we also had those lowercase letters, like I mentioned earlier on. These lowercase letters are used when we are listing the genotype to symbolize recessive alleles for particular traits. So whenever we have recessive allele, that typically gets masked over if there is even just one dominant allele from another parent. So the only time that we will see the phenotype for a recessive trait is when we inherit that, when we inherit those alleles, recessive alleles, from both parents. So you would need to, in this case that I used as an example, inherit lowercase g to see that recessive trait. And one of the examples when we discussed this in the class was comparing the pea pod color, and we noticed that the green color was a dominant color. And only time that we saw a yellow color for the pea pod was if that was inherited from both parents. So it's much more rare to see the yellow pea pods than it is to see the green pea pods. So all of these should be reminders of the concepts that we have talked earlier on in the class. So I hope that they made sense and they kind of came back to you rather easily. If not, you can always go back to the lecture recording. You can always go back to the lecture videos, which then go into a little bit more detail of these processes. One more concept that I really want to emphasize when we discuss this is the concept of homozygous and heterozygous alleles. So I'll just fix that to our screen here. I've been a little bit too rushing. So that slide looks better. So when we have homozygous alleles, that means that we have two of the same alleles for that particular trait. So let's have a look of that a little bit. So in this example where we looked at whether you have straight hairline or whether you have a widow's peak on your hairline. So here we see widow's peak, here we see straight hairline. What we saw that the widow's peak was a dominant allele. So as long as you have at least one capital W, you will see a widow's peak. So example of that would be when we have capital W and capital W, we would see a widow's peak. When we have capital W and lowercase w, we would still end up seeing this widow's peak because just one single dominant allele was enough for us to see that. Only time we saw the recessive trait, which was lowercase w was when you inherit the trait for that straight hairline from each of the parents of yours. So when we are talking, rather than talking about capital or lowercase letters, we can simplify this by using terms homozygous and heterozygous. If something is homozygous, it means that we have two of the same alleles. This can be two dominant alleles or two recessive alleles. So two of the same. Both of those alleles are the same. The time when we talk about heterozygous alleles is when one of the alleles is different from the other one. So in this case, it will be only when we have capital W and lowercase w that we can say that now we're seeing a heterozygous combination of these alleles. Otherwise, if they were the same, it was going to be the homozygous. So now we get to the first question. And anyone who's wondering about how to do the attendance today, I will be taking the attendance based on the completion of the lab for this week. So you have all the way until the lab has been scheduled to be due to get your attendance for this evening's class. So here I have listed a bunch of different genotypes, I think 6 of them in total. And I'm asking you to identify whether this genotype listing that we see for each, whether it's going to be heterozygous or whether it's going to be homozygous. And remember, heterozygous was when you had two different ones, and homozygous when you had the two of the same. So the first one that we see is capital A, capital A. And that will, of course, end up being homozygous because we have two of the same. Both of these alleles are dominant alleles. The next one that I have is actually a mixture. So we have one capital B and one lowercase b. So these are different. It's going to be heterozygous. So in this combination, remember, hetero stood for two of the different kinds. Homozygous stood for two of the same kind. Well, let's have a look at the next one. Here I have capital C and lowercase c. This would lead us to recall that, okay, they are different alleles. One is dominant, one is recessive. So we're talking about heterozygous genotype. Same goes for the next one where we have capital D and lowercase d, again heterozygous. What you'll notice that is that we usually write these with the capital letter first and lowercase letter second if we're talking about heterozygous alleles. Even if the order would be the other way around when you're working out this genetic makeup, we usually end up always writing it out so that the capital letter is listed first. That's just shorthand, and that just kind of makes it easier for us to work through this, but it would be same as in this case lowercase b capital B, or same as we would see in this case lowercase c capital C, or in this case we would see lowercase d capital D. All of these stood for the very same thing. Now let's have a look of the question number E. So in this one I have two of the same. both are recessive alleles. So whenever it was two of the same, we talked about the homozygous genotype. And again, it could be also two of the same as capital letters, as we saw on our very first question, AA. So capital A, capital A. The same goes for capital F, capital F. So this is a homozygous two of the same genotype. I hope that that makes sense. Really, that was just an activity that I've included into the lab for you to get a chance to get practice of making sure that you identify these. The next question allows us now to apply a little bit of everything that we've learned. So I've listed there 3 different genotypes, and I'm asking you to work out what the phenotype, so the final appearance of this trait would be. Remember that genotype was the genetic makeup, which we see for each of these cases, whereas phenotype is going to be what it is going to appear as, what the outcome that we can visually see is going to be. And here you need to have a little bit of background knowledge to be able to solve these 3 genotypes and work out the phenotype that we see. And the background knowledge that I'm going to share with you is that purple flowers are much more dominant than white flowers. So whenever you have an allele for a purple flower, you will, even if it's just one, you will see the outcome of a purple flower. Only time that we see white flowers is if we have 2 recessive genes for the flower color. So since the purple is dominant, we're going to be symbolizing that with the letter, capital letter P. If we are talking about recessive trait, now we're going to use lowercase p because that was the shorthand that we used, lowercase always for the recessive trait, capital for the dominant trait. And remember, all you needed was one dominant allele, and you would see the dominant trait appearing. So let's work through all of these 3 scenarios together. So here we have 2 dominant alleles. So we're homozygous, dominant here. So that will, of course, result in flowers that are going to be purple in color. On question B, we're seeing a heterozygous combination for the flower color. So there's going to be one capital P for the dominant trait and one lowercase p for the recessive trait. Remember that as long as there was at least one dominant allele, that's the characteristic that we will see. So we will again end up seeing purple flowers. Only time that we really end up seeing white flowers is if we have a homozygous recessive pair of genes or genotype. So here we have 2 alleles, and both of them are lowercase p. That tells that both of them are coding for a recessive trait. So we will now see white flowers. So I hope that that application of these concepts and these very, very core concepts that we have learned in the class starts to make sense. And that brings me to third quiz question. And after this quiz question, we're going to take a little break of looking a little bit more theory, and then we'll continue. But I want to ask here for us to apply now other way around the information. So now we actually have been provided information about the phenotype of an individual. So what we would see. So we see this trait, this is the phenotype, but we do not know what the genotype for that individual would be. So we're left with a question mark for the genotype. We will need to work out the alleles that could be behind for this particular phenotype that we see. And to do that, I provided a little bit more information. So this time we're looking at the hair characteristic, whether it's straight or whether it's curly. And straight hair is going to be the dominant characteristic. So that's going to be marked with a capital H. Curly hair is a recessive trait, so a recessive allele. So we will see that only when the allele is lowercase h. So let's list all possible options. And here I do want to remind you, when you're completing your lab, that bear in mind that there might be multiple options for a specific trait. So let's start with the straight hair. So to see straight hair, we need to have inherited one dominant allele at least. It can be that we have inherited both of the alleles being dominant. So in this case, it would be homozygous dominant. So capital H, capital H. Or we can have only inherited one dominant allele, and the other one can be anything else. It could be also recessive, and still we would see straight hair. So this would be heterozygous combination of these alleles. Only time that we're going to see the phenotype of a curly hair is if we have homozygous recessive alleles. So two lowercase h's. So I hope that this makes sense. And do remember that when you're completing these labs, if there are multiple options, I would be asking you to list all those possible options. I've tried to be very clear when you have fields where you type in answer, that you type in only one answer to that, and then there's usually or next to it. So you get to type it twice. The order in which you write it doesn't matter, whether it's capital H, or whether it's capital H, lowercase h, and then the capital H, capital H. So you can do it in any order. So that brings us to very same answers that we just saw when we worked through this together. And this brings us to do final recap of the content that I wanted to discuss with you. And what I want to discuss with you is the concept of the Punnett Square. And again, I assume that this will be familiar to you from the past of things that we have discussed. So Punnett Square was simply a tool that we use. And we use this tool in genetics to work out all possible outcomes of a cross between two individuals. So we're basically predicting what kind of offspring these two individuals can produce. And by predicting all possible outcomes of the offspring, we can then tell that how likely it is that a particular type of offspring results. So what you see here on the right-hand bottom corner is an example of a Punnett square where both parents on this particular case were actually heterozygous. So they had one capital, one lowercase, and we crossed this combination of parents who were heterozygous for this trait. And this trait that I've illustrated here is going to be the pea pod color. So if we had at least one dominant allele, we always saw the phenotype as a green one. And how you prepared these Punnett squares, remember, is that you separated. So for both parents, we had this capital G, lowercase g, and you separate each of these possible alleles into different rows or columns, and then you drew together from both to get a potential mixture that you could get from these parent alleles. So I've done that only for the first and last box on this diagram, but I hope that that makes sense. And we'll get to have a little bit of a practice of making sense of it. So on this particular question, I'm asking you to complete a cross for parents which other one is heterozygous and other one is homozygous recessive. And this trait that we're going to be looking at is the seed surface texture. So round seeds are dominant, so they're going to be marked with capital R, and wrinkly seeds are recessive, so they're going to be marked with lowercase r. So the very first thing that I think we should do, we should map out these parents. So parent A and parent B, and have a look of, they had 50-50 chance of donating each of their alleles. Which kind of allele combinations could they be donating? So of course, for this parent that we're seeing here, we should be seeing, hold on a little bit, we should be seeing here a lowercase r. I think that my automated spelling doesn't really allow me to put what I wanted to put there. So let's fix that quickly. Sometimes computers are a great help, and sometimes they make our life a little bit more challenging by thinking that they know better than we do. So let's have a look of here. This first parent, parent A, as I've labeled that, this parent is going to have the alleles that are heterozygous. So the capital R is going to go to one of the boxes, and lowercase r is going to go to the other one. For the other one, this parent carried homozygous recessive alleles, so it's going to be lowercase r that's going to go to both of these boxes. And now we simply start working across from all of these potential scenarios. So for the first box, we're going to get a heterozygous outcome, so capital R, lowercase r. Second box, we're going to get lowercase r and lowercase r. So that's going to be homozygous recessive. Then let's have a look of the third box. We're going to get one lowercase r, and we're also getting one capital R. So that's going to be our heterozygous. And finally, for the last one, what we're getting is both lowercase, so homozygous recessive outcome. So kind of a little bit tidier write-up from what we just did. We are getting a mixture of 50-50 chance of getting a phenotype that appears as rounded seeds, and then the other 50% is going to be wrinkly. The genotype is going to be 50-50 for heterozygous versus homozygous recessive. So again, the key is going to be that you learn how to produce the Punnett Square data, and you learn how to read it. And you remember that there is a big difference between genotype, which was the combination of letters, and phenotype, which is actually what we see, which I've drawn there for you. So now for the last question on this quiz, I guess that I'm asking you to form a cross where you have heterozygous parent mixed with heterozygous parent for the seeds. It's the trait of the seed structure, seed appearance. So we already have been provided here the cross for the genotype. And the genotype is, of course, going to be our different combinations that we can have. So we can have a homozygous dominant, we can have a heterozygous, and we can have homozygous recessive. And what we'll notice that while we have 1 for the homozygous dominant and 1 for the homozygous recessive, we actually have 2 for the heterozygous outcomes. So it's going to be 25% of a chance of getting this genotype, 25% chance of getting this genotype, and adding these together, it's going to be 50% chance of getting this genotype. Ooh, and I didn't mean to hide that. Sorry about that. My screen is getting a little full here. So that was simply our genotype. We are, however, being asked to report something else than the genotype. What we're being asked to report is the appearance, so the phenotype. So will it be round or will it be wrinkly? And let's have a look of what kind of outcomes we will be ending up getting. So remember, we're again focusing on phenotype. As long as there's even 1 capital letter, 1 dominant trait, that's what we will always see. So 3 out of the 4 outcomes that we saw were going to be round. And only 1 out of the 4 outcomes that we saw was going to be wrinkly. So for the phenotype, 75% is going to be round and 25% is going to be wrinkly. Compare this to what we learned for the genotype. For the genotype, it was 25 for the homozygous dominant, 50 for the heterozygous, and 25 for homozygous recessive. So you'll notice that the phenotype and genotype are very different things. And of course, the answer for this quiz question would be reflected, we can reflect it in terms of proportions or in terms of percentage values, and it aligns with what we just discussed for the phenotype. And that really completes everything that I had planned to cover with you. I'm sorry that I can't meet you tonight in person, but I believe that this lab really is largely just application of everything that you have learned so far. So you should have a very good grasp of what to expect and how to move ahead. And if you get stuck at all, do not hesitate to drop me a message and I'll be more than happy to take time to answer to that, and we'll figure out whether we can figure it out over writing or if we need to do a little bit of an in-person meetup. But that's all that I prepared for tonight. Again, I appreciate you joining me this time kind of asynchronously, and I hope you have a very good day of tomorrow on the Thursday for the Veterans Day. Thank you for your service for anyone who's a veteran or who has family members who are veterans. I appreciate you and I hope you have a good rest of the night. Thank you. Bye.